Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. Now, Chapter 11 has been all about the control of genes, and the topic today is one that if I were planning a class or writing a biology textbook, I wouldn't necessarily put in this chapter, but we're going straight through the book that I'm using, so today we're talking about cloning plants and animals. As always, right now you should see the terms and definition slides popping up on screen so you can pause and grab the vocabulary we will be using today. Or if you want my entire slideshow as a PDF, head on over to my Patreon. The link is down in the description below to grab my PDF that includes those helpful terms and definitions for absolutely free. However, if you do decide to support the channel, support the content here, you will get the Bio for Beginners content and any study material at least a day early. And we're workshopping what's in the other tiers as well right now as my schedule with school is starting to wind down a bit. So without further ado, let's get into cloning. So before we get into the new content, here's some things to know and a quick little review of some of the stuff we've gone over in the previous two Chapter 11 videos. So the first thing is that cells in an organism contain all of the same genes. However, most cells only express a very small percentage of those genes. So all of the genes are present, but some are turned off. This then poses the question, do all genes retain the potential to be expressed whether or not they're turned off or on? Let's begin with plant cloning and the lessons that we can learn from that particular area of cloning. One thing that you may or may not know, depending on if you're an avid gardener or if you've ever grown plants from cuttings, so pieces of a mature plant that are then cut off and either put in a rooting medium or just stuck in some water to grow roots and eventually a new plant. That is cloning the original plant, and this shows that there are already these differentiated cells, so cells that have already become a specific type that can then take on new identities. That is the first lesson that we need to learn from cloning plants. Because if you take a cutting from an area of the stem, then you have these cells that have already basically become a specific cell, all of a sudden need to become something else, new roots need to grow, and this whole plant kind of needs to refigure where things are. So in the lab, they have been able to take single cells from one plant. In the example we have here, it's specifically a carrot and its root cells from a carrot plant. And then they can be used to grow many new plants or clones. So a clone is basically an organism that is produced that's genetically identical to the original donor. Again, clones are genetically identical to one another. So it's not the same as having identical twins where they have the same genetics. It has to be from an original donor. So in the case of the carrot, the original carrot plant that gave up those root cells to then produce many new carrot plants, that's the donor, and all of the new plants are the clones. And cells that can produce every single type of specialized cell, so like what we're seeing here in the plants, the one that can go from differentiated to then ending up producing these single cells into the new fully formed plant are known as totipotent. So any cell that can take on the different cell identities is totipotent. And the new plants that are grown from that donor plant are the clones. So if you take a cutting of a lovely plant in your backyard and successfully get it to root, you've just made a clone. One more thing that we need to keep in mind here is that plant cloning is actually very, very common. Not only in your own backyard where you may or may not be taking cuttings to make new plants for the most inexpensive option as far as propagating your garden goes, or in, say, a botany classroom in the lab where you're learning about different ways plants reproduce. I know that that's a pretty common thing in some of the classes that I've taken. A lot of the classes I've taken come from plant sciences and botany. But plant cloning is very common in agriculture, specifically with things like seedless plants. So seedless plants, because they have no seeds, they've been over the years selected to not produce seeds because it's more advantageous for when you eat them. You don't have to, say, spit out watermelon seeds. These plants can't reproduce on their own because the seeds are where the little plant embryos are, and if you don't produce seeds, you don't produce the next generation. So the only way to really get these plants to continue growing and continue being able to produce those seedless watermelons that people love is through cloning. And it's also very common to clone plants that tend to reproduce very poorly in artificial settings, like gardens. So orchids are one particular plant that's pretty common to clone, especially for these retail florists that then sell the plants. It's not that simple to get orchids to breed and produce seeds, so a lot of the orchids you're going to pick up at your local garden center are probably cloned. And then cloning is also used to select for favorable traits in our crops, such as high yield or disease resistance. So agriculture, cloning is a big plus because it allows us to produce the things we want, whether or not those plants are going to be easy to breed or if they can reproduce on their own. So with these lessons in mind from plants that we can get these differentiated cells to then produce a new organism and essentially be totipotent, and cloning can be used to select for things that we want, the next question we need to ask is this possible to clone animals? Well, there is some evidence in the animal kingdom that does suggest that yes, cloning is very possible. And specifically, it's when we think about examples of regrowth or regeneration of lost body parts. So regeneration is just the regrowth of a lost body part. And we can see examples of this throughout the animal kingdom, all the way from simple animals like sea sponges, where you can take a couple of cells of the original sponge, put it somewhere else, and a whole new cloned organism will begin to grow. But sponges are very simple. They don't even have true tissues. So then we can look at animals like the invertebrates. So the example we have here is a sea star that's lost its arm. You can see the one arm is much shorter than the other ones there, but it's regrowing. A fun fact about sea stars is that depending on where they're cut and how much of that central disc is left on an arm, a whole new animal can regenerate. But that's a fun fact, and we're not really talking about our lovely friends, the sea stars, right now. And then all the way up to chordates. So you and me, we're chordates. That's the phylum we're in, but we also share this phylum with so many other animals like reptiles. And I'm sure that everyone knows that if a lizard drops its tail, it's gonna regrow. Now, there is a little asterisk here in that not all species of lizards do this. Crested geckos, for one, can drop their tail, but they never regrow their tail once it's dropped. And the new tail is not going to be exactly the same as the old one that it lost, but this is still regeneration. They're still regrowing a lost body part. Now that we see that there are some examples of at least regeneration in the animal kingdom, we can actually discuss animal cloning. A quick spoiler here is that cloning animals is possible. It has been done. But it's not quite as simple as cloning plants. You can't just walk up to an animal, chop off a piece of their body, stick it in a cup of water, and have a whole new organism sprout like you can in a plant. you need to go through a particular process, and this process is nuclear transplantation. And the only way to explain what nuclear transplantation is to walk you through the steps. So step 1, the nucleus of an egg cell will be removed. The nucleus of a cell, remember, holds all of the DNA and all of that genetic information. And an egg cell has half the possible chromosomes for an organism, because an egg cell is a sex cell, and sex cells are haploid, so they have half the number of chromosomes because they're meant to join with a sperm cell and then form a brand new organism with a brand new genetic makeup. But here, we're removing all of that genetic information from that cell. And then in step 2, we're actually going to inject the nucleus of a somatic cell. So a somatic cell, remember, is just a cell that's not a sex cell. However, it contains all of the DNA and all of the chromosomes necessary to produce a brand new organism. Just in this case, the actual genetic makeup in this nucleus belongs to the donor organism. So the nucleus we're using comes from the donor, not necessarily the egg that we're using. That's the big thing to remember here. The donor gives us the nucleus. And so then that cell with that brand new DNA injected into it is going to be stimulated so it can form what is known as a blastocyst. Now, blastocyst is basically just a really early embryo. It's a little bit more nuanced than that, but for our purposes, just think of it as an early embryo. And once that blastocyst is formed, it can then be implanted into a surrogate mother, and eventually a new animal, which will be the clone, can be born. And this specific process is known as reproductive cloning because that blastocyst is implanted in a surrogate mother and then born. So reproductive cloning, you're going through part of the reproductive process. It's just not joining an egg and a sperm. So this is just a little bit better of a blow up of that figure we had on the previous slide that shows you all the steps we just talked about. And feel free to pause and look at it again. The steps are exactly what we just went through. So a little bit of the history of animal cloning. In the 1950s, nuclear transplantation was successfully performed for the first time using cells from frog embryos. Now, in the following decades, there was some success cloning mammals with very young cells from different embryos. But probably the most famous cloned animal that you will hear about is from 1996. So in 1996, the Scottish researcher Ian Wilmot and his team cloned a sheep known as Dolly. Now, to give you a little bit of a perspective of how difficult this whole cloning process is in the animal world, during the process of giving rise to Dolly, they successfully created 277 zygotes that resulted in 29 implantations and 1 live birth. And Dolly did pass away at the age of 6. She had complications from, I think, a lung disease that's actually much more common in older sheep. And for her particular species, life expectancy is 12 years old. So that was one thing. She didn't survive as long as you would expect a sheep to survive. And so this brings us to the question, why even clone animals? Well, one thing is disease research. Genetically identical animals actually help increase control in testing for certain things, because then that takes away the question of, was it a difference in the genetic makeup of animal A and B that caused the result we're seeing in our test? If you have a bunch of clones, that controls for the genetic factor. It also has the potential to improve agriculture in the way that cloning plants does, because then you can select for desirable traits. Like in sheep, you can easily just have a couple of sheep that produce much better wool than other sheep. Well, clone them. Or you could do that with milk production or better beef or whatever. That is one of the big question marks above this particular area of discussion. Since cloning has improved agriculture in the plant world, it is possible it could in the animal world as well. The production of pharmaceutical drugs is one reason people are still very interested about cloning animals, because there are some drugs that come from different animals, and cloning them will increase the supply. The last point where the kind of why clone animals is a big deal is when you talk about the conservation of rare, endangered, and even some extinct species. So there's a frozen zoo in San Diego, California that currently houses the cryogenically preserved samples from 900 endangered animals. Most famously, one of the species that may actually be helped through cloning and things like that is the northern white rhino. At time of recording, there are only 2 northern white rhinos left in the world, but at the San Diego Zoo, they've been working on essentially hoping to use southern white rhinos as a surrogate to help raise new northern white rhinos. Now, there's a big question mark about whether or not this will be successful, but they have at least had some success with artificial insemination of other southern white rhinos into their lovely southern white rhino girls that have been selected to hopefully be the champions for their northern cousins. But I digress on this point. So what are the problems with animal cloning exactly? Well, as of right now, clones tend to have health problems and die young. As we touched on, Dolly the sheep passed away at age 6 when her life expectancy is about 12. And researchers are currently trying to solve this problem. One of the current hypotheses is that because the DNA is coming from older organisms, the new animal when it's born is, at least on its DNA clock, kind of older than a newborn. So it's a bit more complicated than that, but they think that if they use the youngest cells possible as the donor DNA, that will help to kind of fix the problem, as well as looking at the methylation of the DNA and different tags that make the whole animal seem older than it actually is. But that's still a big area of research that has a lot more questions than answers at the moment. And cloning is also hard and expensive, so there's a question of, is it even worth it? Because remember, with plants, it can be as easy as taking a quick cut and plopping that little cutting in a glass of water and waiting for roots to form. In animals, it's a much more involved process and it's much more expensive, so you kind of have to weigh the rewards and the costs to determine if it's going to even be worth the whole process. And then there's this big question of, is it ethical to clone animals? And that is something that's still very hotly debated, and that's a question we're not going to get into here, because quite frankly, when we're having a discussion about whether or not it's possible and how it's done, we're not talking about ethics here, we're talking about the biology behind it. So for this video, a discussion of the ethics is not relevant. Now we have one more cloning thing we need to talk about now that we've gotten over cloning plants and cloning animals. And this other thing is something known as therapeutic cloning. And so therapeutic cloning is essentially where you take stem cells of some sort, usually embryonic stem cells or ES cells, as you'll see them sometimes abbreviated, to treat a disease. So what exactly are embryonic stem cells? Well, they're undifferentiated cells. So they're a cell that can still take on any identity. They haven't taken on an identity yet, so that's what undifferentiated means. And they can divide pretty much indefinitely in a laboratory culture because they have no stimuli to force them to take on an identity. And they come from a blastocyst or an early embryo. So every single animal, every single organism has embryonic stem cells at some point in their life cycle. And then there's also adult stem cells that can be used for therapeutic cloning, but the embryonic stem cells are the best because they're entirely undifferentiated. Adult stem cells, on the other hand, are partially differentiated and they're found in bone marrow. So they can only take on some identities, specifically blood cells. So they can be very useful in treating like immune diseases. Now, it's important to note here that therapeutic cloning is still being researched and success is still considered rare, at least at the time of filming this video. And using embryonic stem cells is highly controversial, and that's one of the reasons therapeutic cloning is still kind of in its infancy. So that's pretty much as in-depth as we're going to get with therapeutic cloning. As you can see here, this figure has kind of an idea of sort of what they do. So the embryonic stem cells can be cultured in the lab, and then they can be used to treat things in like the blood cells, so the immune system, in nervous cells, because they can take on these identities we see here. They can be used to treat diseases and disorders in the immune system, in the nervous system, in the heart system, in the muscles. But therapeutic cloning, again, success is kind of hit or miss, and it's still in its infancy. And it's very, very hotly debated because of the embryonic stem cells are the best cells to use for it. Because as we can see in this figure, if you do use the adult stem cells, you can really only use them for taking on a blood cell identity since they are already partially differentiated. But that's all I really have for you today. Again, I would not necessarily have put cloning in this chapter's discussions, but we're just following the book, how it's laid out. I'm not making a lesson plan or a course really here. So we are at the book's mercy, and I hope to see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.